Good morning, students. Today we are having lecture number 15 in microbiology, and today we will talk about the causative agents of enteroviral and neiroviral infections, and we also will discuss causative agents of hemorrhagic fevers. So this lecture includes those viruses that cause viral infections of digestive system, that infect organs of digestive system and central nervous system. That's why they are called neiroviral infections. And another group that will be discussed includes viruses that cause hemorrhagic fevers. I remind you that hemorrhages means bleeding. It means that these infections are characterized by occurrence of bleeding. So let's start. And the first virus that we are talking about now is polio. It is the causative agent of poliomyelitis or polio. Characteristics of virions, antigens, and cultivation. You remember that viruses are non-cellular obligate parasites. They have no cell organization. Viruses are distinguished from bacteria because bacteria are cellular organisms. Viruses are non-cellular organisms. That's why description of viral infections and viruses is distinguished from description of bacterial infections that we used earlier in lectures about bacteria. So, the causative agent of poliomyelitis. It belongs to family Picornaviridae, genus Enterovirus, and species poliovirus. It is the causative agent of poliomyelitis. We also can call it polio. So disease that is caused by poliovirus is named poliomyelitis or polio. In this diagram, in this photo, you can see image of ancient Egyptian with polio. Look please in this photo. It means that polio is ancient disease that was known from ancient times, and a lot of people were infected with polio. And we can see that this man has a main clinical manifestation of polio. If we compare this leg and this leg, so what happens in polio? What is this virus? Let's talk about it. Firstly, about morphology. Capsid of poliovirus contains about 60 subunits. Each subunit consists of 4 proteins. They are called VP1, VP2, VP3, VP4. VP means viral protein. So one subunit, it is one subunit. It consists from 4 viral proteins. And such subunits are included in structure of poliovirus. There are about 60 subunits in viral particle. This virus have single strand RNA. It is positive chain. I remind you that what does it mean? RNA positive or RNA negative? RNA positive viruses have RNA positive chain of nucleic acid that play a role of messenger RNA. It means that protein synthesis of these viruses occur in ribosome of host cells, and RNA positive chain also plays role of messenger RNA. It serves, it saves genetic information, it stores genetic information, and it also transfer this information into ribosomes of host cells when there is a replication of poliovirus. Unlike RNA negative chain, when messenger RNA must be produced, and information is transmitted from negative RNA chain, RNA minus, to messenger RNA, and just after it into ribosomes of host cells. We will remind you about it at practical class. There are about 3 antigenic types of polioviruses based on epitopes found in the antigens VP1, VP2, and VP3 proteins. Cultivation of poliovirus. Poliovirus can be cultivated on kidney cells of green African monkeys and on diploid human cells. How to understand that viruses replicate in these cells? We can see cytopathic effect. What is it? What is cytopathic effect caused by poliovirus? Cytopathic effect is attended by destruction and the formation of granules in the infected cells. So we can compare non-infected kidney cells of African green monkeys or diploid human cells and infected. And we can understand that poliovirus is present in specimen if we can see destruction of these cells, destruction, formation of granules in these cells. Epidemiology and pathogenesis of poliomyelitis. The first one, what is cause disease? I have said about it sometimes. It is polio. Caused disease is called polio or poliomyelitis. You may call it by both names. Source of infection are sick men, sick human. It's mostly they are mostly sick children, because polio infect human in most cases in the childhood. It may be apparent and subclinical patients. We will talk about different stages, different clinical forms. So we know that there are apparent clinical forms when we can see clinical manifestations of polio, but in some cases, and it's in most cases, these are subclinical forms. Transmission. The first transmission way is a fecal-oral transmission. What is it? Please, when you learn this lecture, you should understand what is it, and you should know what is it. You should explain. Fecal-oral transmission means transmission of poliovirus due to poor hygiene, due to dirty diapers, because it's child's disease, especially in daycare settings, through ingestion via contaminated food and water, and contact with infected hands. And another transmission way is airborne, through inhalation of infectious aerosols. If we come back to history of polio study of poliomyelitis, scientists and doctors studied polio and transmission of polio during a long period. They made a lot of experiments with animals. They observed a lot of children in their families to understand how is it transmitted. And the first transmission way that was discovered, it was fecal-oral. And just after some time in experiment, there was shown that polio is also transmitted by airborne transmission. It was an experiment with chimpanzee. Scientists dropped culture of viruses intranasally in nose, and it caused development of polio. So they proved that polio also transmitted through airborne transmission. Incubation period of polio takes about from 1 week, from 7 days up to 14 days, up to 2 weeks. Pathogenesis of polio. We have just said that poliomyelitis is a neuroviral infection. Neuroviral means that this virus have tropism to nervous cells. Tropism means when virus infect, what are target cells for this virus. These are nervous cells. How disease develops? Firstly, virus enters the mouth of the child. Then it multiplies in tonsils and lymph nodes of the neck. If it is transmitted by airborne transmission and it multiplies in pair patches, I hope you remember that pair patches are located in small intestine, in mucosa of small intestine. It's secondary immune organs, lymphatic organs in small intestine. So if virus is transmitted by airborne transmission, it multiplies in tonsils and lymph nodes. So in cells of reticuloendothelial system, I hope you remember that reticuloendothelial system includes lymphatic cells, multiple lymphatic cells. And in pair patches, if it's transmitted by fecal-oral transmission. Then viremia occurs. Viremia, it's circulation of viruses in the bloodstream. Then viruses reach axons of peripheral nerves and they are transferred to the central nervous system along these nerves. They infect peripheral nerves and then they reach central nervous system through moving along these nerves. Then they move along the fibers of the lobar motor neurons into spinal cord or the brain, and they infect motor neurons. They infect nervous cells, neurons of spinal cord and brain. They invade nerve cells and cause their destruction. Then it causes intracellular multiplication of viruses, intracellular multiplication of viruses in motor neurons, in spinal cord cells and brain cells, and it causes destruction of motor neurons in the spinal cord. Motor neurons and cells are destroyed and it causes flaccid paralysis. Flaccid paralysis due to inability to cause muscle contraction. Motor neurons, you remember that motor neurons are combined to muscles. They are destroyed, so it causes inability to muscle contraction. It is flaccid paralysis. I hope you remember that we studied another disease, bacterial infection, that also caused flaccid paralysis. What is this disease? It is botulism. But pathogenesis and action of botulism is distinguished completely from pathogenesis of polio. So, there are three clinical forms of polio. The first one is inapparent infection. It's infection without any clinical symptoms. It's also called asymptomatic illness. Inapparent or asymptomatic infection is characterized by non-specific symptoms like fever, malaise, drowsiness, headache, nausea, vomiting, constipation, and sore throat in various combinations. Sometimes we can see just fever with headache, sometimes we can see vomiting, only vomiting. So it is inapparent infection when we can't see paralysis. Another clinical form of polio is called non-paralytic poliomyelitis. Non-paralytic poliomyelitis, or it's also named aseptic meningitis. Aseptic meningitis. In addition to the above symptoms and signs that we have just talked about, fever, malaise, drowsiness, headache, and so on, the patient presents stiffness and pain in the back and neck. So the patient with non-paralytic poliomyelitis has fever, malaise, headache, nausea, and so on, and he or she, this child, also has stiffness and pain in the back and neck. Why does it happen? This form is also called aseptic meningitis. Aseptic because it doesn't cause sepsis without purulent focuses in the brain, but it is meningitis. It's inflammation of meninges in the brain. Meningitis is characterized by such clinical symptoms as stiffness in neck. When we ask the patient to move or to touch with head his knees or his leg, for example, it is impossible for the patient because it causes muscle contraction due to severe pressure of cerebrospinal fluid in neck, and it causes muscle contraction. Another clinical form of polio, it is paralytic poliomyelitis. Paralytic poliomyelitis. It is a clinical form that is characterized by specific clinical forms, by visible clinical forms that is specific for the disease. It's flaccid paralysis resulting from lobar motor neuron damage. Flaccid paralysis. Let's look at these photos. In this photo, we can see result of polio in the child. You can see this girl with paralysis, flaccid paralysis. What is the result of flaccid paralysis? It is atrophy of muscles. Muscles are atrophied. That's why such child never able to walk normally, and the muscles never remain normal. In these photos you also can see clinical manifestations. It's flaccid paralysis when the child is unable to walk themselves. And here you also can see clinical manifestation of polio. The children who had polio in the childhood became disabled persons or invalid persons for all the life, because it's direct destruction of motor neurons. Because of direct destruction of motor neurons, that cause full atrophy of muscles. Microbiological diagnosis, treatment, and prophylaxis of polio. Specimen. It includes stool collection, because we know that virus multiply in small intestine, in parapatches cerebrospinal fluid, because virus reach central nervous system, and oropharyngeal secretions, because virus also are transmitted by airborne transmission. Methods for detection includes using of PCR, then detection of antigen in specimen. For example, it's a neutralization test. The virus isolation or cultivation virus, I have said how can we cultivate it. And serological tests that include ELISA and complement fixation test that are used for detection of antibodies in serum of the child. What about treatment and prophylaxis? Firstly, about prophylaxis. Today, there are obligatory vaccines that are used in all the countries. In all the countries. In immunology, we talk about it that vaccine against polio is necessary. It is used for any child as a necessary vaccine. There are two types of vaccines that are used. The first one is oral polio vaccine, or its short name is OPV. It's a live attenuated vaccine. That's why it is dropped orally, per orally. It was made by Dr. Sabin in 1957, from this year up to 1962, when he worked under the development of this vaccine. And an activated poliovirus vaccine is another one, or IPV, that was made by Salk. Inactivated poliovirus vaccine is injected intramuscularly. This vaccine was prepared, was made earlier than oral polio vaccine. Actually, creation of vaccine was a great step in medicine, because a lot of children, you don't imagine, a lot of children were or became disabled persons. Polio, it is a disease of developing countries, of all developing countries. And the doctors made experiments with their cells to check effectiveness of vaccine. What about treatment? There is no specific treatment. There are no antiviral drugs for treatment of polio. Treatment involves reduction of pain and muscle spasm and maintenance of respiration and hydration. The immunity after polio is lifelong, and it's very important that there is a passive immunity in polio. It's transferred from mother to newborn. The maternal antibodies gradually disappear during the first 6 months of life. Actually, if mother is vaccinated before pregnancy, or if mother was infected, for example, polio in the childhood, in her organism there are antibodies that are transferred through breast milk. That's why the first 6 months the newborn is protected via these antibodies. And vaccination against polio, the first vaccine injection is given after 6 months. What else should we say about polio? I have said that today polio is prevented in all countries. It's necessary vaccine in all countries whenever you will work. A major campaign was launched by the World Health Organization in 1988 to eradicate poliovirus from the world. There were an estimate about 350,000 cases of polio worldwide in 1988. Americans were certified as free from poliovirus in 1994. The Western Pacific region in 2000, and European countries, they said that they finished polio development, polio spreading in their countries in 2002. In 2014, only 3 countries, Afghanistan, Nigeria, and Pakistan, remained polio endemic. It means that places that some maybe poor towns, poor small towns, or villages where there are places with polio were left. And sometimes we can't be sure that today polio is eradicated completely. No. In some African countries, you know that not all children are vaccinated. There are people who live in very, very poor regions where they don't know about vaccination. What about India? India was certified as polio-free in March in 2014. It also occurred due to widespread vaccination. Outbreaks of poliovirus sometimes occur in previously polio-free countries because of importation or transfer of the virus by travel and migration from those countries where it's not used, or from these poor regions. The another pathogen that causes infection of central nervous system, it is a causative agent of rabies. It belongs to family Rhabdoviridae and genera Lissa virus. It is the causative agent of rabies. You can see on these photos, and you may understand what are the main sources of rabies. Firstly, about morphology of the virus. Lissa virus that causes rabies has bullet shape. You can see it resembles bullet. Its genome is RNA negative chain. It's enveloped virus, so it has envelope. Its antigen includes nucleoprotein, this one, and G glycoprotein, this one, that is major factor in rabies virus neuro-invasiveness and pathogenicity. So this surface antigen is responsible for invasion of nervous cells. There is a single serological type of a rabies virus. What about cultivation of rabies virus, of Lissa virus? Available tissue is inoculated intracerebrally into suckling mice. Available tissue means tissue with viruses, with Lissa virus. Tissue, nervous tissue from infected person or animal is inoculated intracerebrally into suckling mice. You can see suckling mice are newborn mice. We also can cultivate it in chick embryo, in diploid human cells, and in vero cells. And diploid human cells and vero cells are used for cultivation of viruses in vaccine production. I will remind you about it once more when we talk about vaccine. Epidemiology and pathogenesis of rabies. What about source of rabies? There are urban forms of rabies and sylvatic forms. Urban means that is transmitted from animals who live next to the people, who live together with people. These are dogs and cats. And sylvatic forms, sylvatic or wild forms, we can say wild forms. These are wild animals, like bats, foxes, raccoons, wolves, skunks, coyotes, mongooses, and other biting animals. And the only transmission way through by animal bites, biting of animals. It is known. You know that normally wild animals like foxes or raccoons or wolves, they are afraid of human. They never come to the human because they are afraid human. It's not dogs, domestic dogs or cats that love human. But when wild animal may come to the human when they are infected with rabies and when this virus destroy all nervous cells in the brain. That's why wild animals lose this feeling. They lose fear of human. That's why they come to the human and can bite the human. And in this stage of the disease in animal, viruses are present in large amount in saliva, because from the brain the virus enter into such human body fluid as saliva. What about pathogenesis? Firstly, I have said to you that it's transmitted by biting of animals. Virus enters tissue from saliva of biting animals. In saliva of animal infected with rabies, there are a lot of viruses, a lot of lyssavirus. So virus enters this tissue in biting sites with saliva. Then virus in the second stage replicates in muscle near biting sites, near bite. Then lyssavirus have tropism to nervous cells, so it invade peripheral nerves due to G-glycoprotein. Virus moves up peripheral nervous system to CNS, moves up. Then virus ascends spinal cord invade nervous cells of spinal cord, motor neurons. Virus reaches brain and causes fatal encephalitis. After that, virus enters salivary glands and other organs of victim, other internal organs. Incubation period of rabies is average from 3 up to 8 weeks. I want to come back to this scheme, of pathogenesis. Why is incubation period different in pathogenesis? The timing, the period of incubation period, depends on the site of biting. If site biting is in the leg, incubation period is long, sometimes even more than 2 months. If animal bites neck, incubation period is about 1 week. Why? Because Lissa virus, needs time to reach central nervous system. They say that the speed of viral movement is about 3-4 millimeters per 1 hour. 3 or 4 millimeters per 1 hour. So virus moves to the central nervous system with a speed of 3-4 millimeters. So once more, incubation period depends on the site of biting. If it is next to the central nervous system, it is short. It is far from central nervous system, it is long. There are some phases, 2 phases in rabies. The first one is a prodrome phase. It takes from 2 up to 10 days when virus infects peripheral nerves, travels up the central nervous system to the brain. It takes about from 2 up to 10 days. Once more, it's also may be different due to that depends on the site of biting. And the second one is neurological phase. Neurological phase that also takes from 2 up to 10 days. Virus infects the brain, damage motor neurons that results in paralysis. When motor neurons are destroyed, it results in paralysis. It is a general result in rabies and in polio. Because motor neurons are not combined with muscles. Peripheral motor neurons are not combined with muscles. Paralysis of the throat causes drooling and the inability to swallow. The patient is unable to swallow saliva or water or even swallow because it's our reflex, normal reflex. Then we can see such clinical manifestation as hydrophobia. Hydrophobia, it is a fear of water. Hydrophobia is triggered by the pain associated with the patient's attempts to swallow water. I have said that due to paralysis of muscles in throat, patient is unable to swallow. And when patient can see water or can hear that water is a flu, for example, fluing, the patient becomes very stressed because reflex, when you can see water, it's reflex, we want to swallow. But the patient with rabies is unable to do it. The patient also has such clinical symptoms as a focal and generalized seizures, disorientation, and hallucinations due to destruction of brain. The paralysis leads to respiratory failure due to paralysis of respiratory muscles, coma, and death. Microbiological diagnosis of rabies, treatment and prophylaxis. Specimen. A specimen includes collection of tissues, nervous tissues, where viruses are present. It's a biopsy from infected sites in biting because we know that virus multiply in the site of biting, and preparations of brain or cornea tissue. Methods that are used for cultivation, for identification is PCR, the first one. The second one is detection of viruses by detection of Negri bodies. What are Negri bodies? Negri bodies, look at this photo, at this smear. Negri bodies are cytoplasmic inclusion bodies with viral particles in neurons. It is neuron in cytoplasm of neuron. We can see inclusion bodies and viruses are present there in large amount. So we can prepare smear from nervous tissues that contain neurons and stain it, and then we can see Negri bodies. And another one that is used in diagnosis of rabies, these are serological tests, immunofluorescence test and neutralization test. Actually, it is very important to understand that after biting of wild or domestic animal, we must not wait for result of diagnosis. Mortality rate in rabies is 100%. Any person without vaccination die. So, no effective treatment exists in rabies. After biting of both wild or domestic animal, the patient must immediately undergo post-exposure prophylaxis. It means a series of anti-rabies vaccine and immunoglobulin injection. We must not wait for results of diagnosis. We don't know this animal is infected with rabies or is not infected with rabies, but post-exposure prophylaxis means injection of vaccine in any person who is bitten. There are two types of anti-rabies vaccines. These are human diploid cell vaccine, HDCV. It's called diploid because they create this vaccine by cultivation of virus in diploid cells. And another one is a purified chick embryo cell vaccine, PCECV, purified chick embryo cell vaccine, because they cultivate virus. They created this vaccine by cultivation of virus in chick embryo. These vaccines are administered in a series of some injections on the first day. The first day means right after biting of animals. On the third day, then the 7th, the 14th, and the 28th day. Some doctors advise to observe, if it is possible, observe the animal. If animal doesn't die during 10 days after biting, it means that we can stop other injections, left injections, because the animal is healthy. Sick animals that are infected with rabies die during 10 days after biting. They die because at this time when wild animals bite the human, at this period their brain almost is damaged, it's destroyed. So in any case, just some days are left for the life of wild animals who bite the person. If a person is bitten, the wound should be thoroughly washed with soap. The first medical care that you may do for the patient in the absence of vaccine, the first that you must do, it's washing of the wound with soap. It's known that soap inactivates virus during 15 minutes. So you should wash it with soap during 15 minutes. And I have said that mortality rate in rabies is about 100% without using of vaccine. Any person who is infected with rabies die if we don't inject vaccine. Vaccine in rabies, it's very interesting because we used to say that vaccines must be injected before infection, before appearance of infection in human. But I have said to you that virus enters and move up to the central nervous system during long period, very slowly. That's why we have time during it. This time is enough to produce cellular and humoral immunity to fight, to protect this person from the destruction of nervous cells. We don't know what about immunity after rabies. Why? Why? Why we don't know that immunity is lifelong or short-lived? Why? Because nobody is alive after rabies. Nobody is alive. Another group of viruses that we will talk about today are arboviruses. Arboviruses, it's not name of a family, it's not name of genera. Arboviruses is a name given to arthropod-borne viruses, that are viruses transmitted to people and mammals by blood-feeding insects called arthropods. These are arthropods. Arthropods, they transfer virus from mammals or from sick human to healthy person. Actually, insects or arthropods never source of infection. They are only vectors. Please remember it. That ticks, for example, or mosquitoes are never source. They are only vectors. They only transfer this virus from one person or animal to another. And the first group of viruses that we talk here, these are flaviviruses. Flaviviruses. Flaviviruses belong to family Flaviviridae, genera Flavivirus, and flaviviruses cause such diseases as tick-borne encephalitis. Another name is Russian spring-summer encephalitis. Russian spring-summer encephalitis. The second one is dengue fever, and the third one is Omsk hemorrhagic fever. Please call it correctly. Omsk, not OMSK. It was a great mistake last year, pre-last year, students made this mistake. Omsk. Omsk is a city in Russia where it was discovered. Omsk hemorrhagic fever. Flaviviruses is named due to the first disease that was discovered. Yellow fever. Maybe you know about yellow fever. It was the first disease that was discovered caused by flaviviruses. Yellow in Latin language means flavi. Flavi is yellow, translated as yellow from Latin language. What about morphology of flaviviruses? They are spherical viruses, you can see, they are spherical viruses. Their genome is a single-stranded RNA that is positive chain. It means that RNA store genetic information and transfer genetic information into host ribosome. It plays a role of messenger RNA and storage of genetic information. This virus is enveloped, it has envelope. There are 3 structural polypeptides and 2 of them glycosylided. What are antigens? It's E enveloped protein and E enveloped glycoprotein. Cultivation of flaviviruses is possible in cell lines, Vero cells and HeLa. Epidemiology and pathogenesis diseases caused by flaviviruses. Firstly, about diseases. I've said that we talk about 3 diseases caused by flaviviruses. It's tick-borne encephalitis, or TB, its short name, Omsk hemorrhagic fever, and dengue. Transmission of tick-borne encephalitis is transmitted through biting of ticks and foodborne transmission from cattle. So, host or source of tick-borne encephalitis are rodents, mammals, wild mammals, or it may be domestic farm animals, cattle, for example. It is spread in European countries and in both Asian countries. The another disease that is caused by flaviviruses is Omsk hemorrhagic fever. It's transmitted by biting of ticks. The source of infection are muskrats. It's widespread in Siberia. Siberia, it's region of Russia, where it was firstly discovered. But it doesn't mean that it is present only in this region. No. It may be present in other regions, other countries too. And dengue fever is transmitted by biting of mosquitoes from human and monkeys. It's widespread in tropic countries and in any country where this Aedes aegypti, it's special of mosquitoes, Aedes aegypti are present. The first one is a tick-borne encephalitis, or Russian spring encephalitis. From the name of this disease, you may understand that it's transmitted by biting of ticks, and it causes the encephalitis in human. Firstly, about transmission. Maybe this scheme is difficult for you, but I'm going to explain. I want to show you that in different stages of development of ticks, they are infected. For example, adult ticks before winter, for example, in late autumn, lay the eggs, produce eggs. If this tick is infected, it means that eggs are also infected. It is called transovarial transmission. Transovarial transmission when laid eggs or produced eggs of ticks are also infected. That's why after winter, they are served in the soil. After winter, larvae that grow from eggs infect rodents. It happens when? It happens in early spring. It happens especially in April and May, when it becomes warm in Russia. Then larvae become nymphs, and it can larvae mostly bite small mammals, rodents, because larvae are small. They live on the surface of ground. They're unable to bite large animals. So they bite rodents. So the source of tick-borne encephalitis may be rodents. Then larvae become nymphs, and nymphs are larger in size than larvae. They may live in the grass, on the trees, then they bite large animals and birds, deer, for example, foxes, and domestic animals that eat grass. That, for example, nymph can bite sheep, cow, goats, and other cattle. So people can be infected. Person can be infected through infected milk. Two transmission ways of tick-borne encephalitis are known through bite of ticks from wild animals. I have explained about it, and through milk, foodborne transmission through infected milk from goat, from cow, through raw milk, or sometimes through cheese. So source are rodents and other mammals, sheep, cows, goats. It's about domestic animals. Incubation period of tick-borne encephalitis is between from 4 up to 21 days. Virus enters the human blood when tick bites. You can see when tick bites the person, it causes primary viral multiplication in lymphoid cells or in vascular endothelium. Viruses multiply in lymphoid cells or in endothelium, in blood vessels. Then viruses are transferred through the central nervous system to the brain and multiply there. Multiplication in the central nervous system, it causes neuronal degeneration, destruction of neurons. Disease starts suddenly with severe headache, chills and fever, nausea and vomiting, generalized pains and malaise. Within 24 up to 48 hours, drowsiness develops and the patient may become stuporous when he or she doesn't understand who is she or who is he. Mental confusion, tremors, and convulsions and coma develop in severe cases due to degeneration of neurons in the brain. If the patient is alive, immunity is lifelong after the disease. In this map, you can see worldwide spreading of tick-borne encephalitis. Risk area of tick-borne encephalitis is it's like belt. You can see like belt. These are all countries, mostly forestic countries where there are forests where exotic ticks live. In Russia, here is Russia. In Russia, the most territory is included in spreading of tick-borne encephalitis. You can see that European countries also included in spreading of tick-borne encephalitis. China also. The another disease that is caused by flaviviruses is dengue, or it's also named break-bone fever. The source of infection are humans and monkeys, and transmission by bites of mosquitoes. I have said it's mosquitoes Aedes aegypti. Incubation period is from 4 up to 7 days. Early signs of infection are sudden high fever, prostration, sharp pain in the muscles and joints. Sensations feeling like their bones are breaking, thus the disease has been called breakbone fever. Disease is named breakbone fever due to severe pain um, in bones, but sure it's not in bones, it's in joints and muscles due to um, destruction of blood vessels by this virus. Dengue hemorrhagic fever is characterized by rashes from skin hemorrhages appears on the face and extremities, and severe vomiting and shock ensue. It's called dengue shock syndrome, as blood pressure decreases dramatically due to destruction of blood vessels once more. Immunity is lifelong against this serological type. If another serological type in another country, it may occur once more. So dengue fever is characterized by rashes in some cases due to destruction of blood vessels and severe pain, sharp pain in bones and joints. What about spreading of dengue in the world? You can see that affected countries that included in spreading of dengue are Brazil, Argentina, Peru, Canada, Mexico. Then these are Asian countries. I think that all students from India know about the disease. It's India. These are mostly tropic countries, Thailand, Malaysia, so Singapore. In those countries where these mosquitoes live, where Aedes aegypti live. Dengue fever is really a widespread disease in these countries, tropic countries. And another disease that is caused by flaviviruses is Omsk hemorrhagic fever. Omsk, once more, it is a city in Russia, in Siberia, that it was discovered firstly. The source of Omsk hemorrhagic fever are muskrats. These are animals that live along the rivers. Transmission ways through biting of ticks, or sometimes through contact with a muskrat, contact with blood, feathers, or urine of a dead or sick muskrat. Muskrats are objects for hunters. That's why hunters are under the risk to be infected via Omsk hemorrhagic fever. Virus cause the destruction of the vascular wall, endothelium, which causes hemorrhagic syndrome and local hemorrhages or bleeding in the internal organs. It also causes destruction of the central nervous system. What are symptoms? These are pain in the lower and upper extremities, severe prostration and rash on the soft plate, swollen glands in the neck, appearance of blood in the eyes, it's conjunctival suffusion due to bleeding, dehydration, and hypertension due to destruction of cells in central nervous system. If we compare three diseases, tick-borne encephalitis, dengue, and Omsk hemorrhagic fever, we can see that the first one, tick-borne encephalitis, infect mostly central nervous system. Dengue infect and mostly blood vessels, endothelial cells of blood vessels, and central nervous system little bit. Omsk hemorrhagic fever infect both endothelial cells, blood vessels, and nervous system, central nervous system. Microbiological diagnosis, treatment, and prophylaxis of tick-borne encephalitis, dengue fever, and Omsk hemorrhagic fever. Specimen. We can collect blood, but only early in the infection. Cerebrospinal fluid and tissue specimens when viruses may be present. Methods for diagnosis is PCR, then direct detection by the cultivation of the virus in tissues, and serological method like neutralization test, hemagglutination inhibition test, and ELISA. What about treatment and prophylaxis? There is no specific treatment for arboviral infections, but there are vaccines for prevention of these diseases. In tick-borne encephalitis, there is a killed vaccine for people who are at risk for contact. For example, who live in the forestic regions where tick-borne encephalitis, who spend a lot of times due to their profession, due to their job in the forests, they must be vaccinated against tick-borne encephalitis. In dengue, there is also live vaccine that was licensed about 7 years ago in 2015. And in Omsk hemorrhagic fever, there is a killed vaccine. Vaccination against Omsk hemorrhagic fever is performed according to strict epidemiological indications. If there is this disease in region. In immunology, I explained what is epidemiological vaccination, if there is a risk of this disease in this region. Another group of diseases that we also discussed today, these are rodent-borne hemorrhagic fevers. Rodent-borne hemorrhagic fevers, you may understand that it's transmitted from rodents, only from rodents. The only group of viruses here are Bunya viruses. It belongs to family Bunyavirida. The name Bunyavirida was given due to the place where it was discovered. Bunya viruses were discovered in town Bunyamwera in Uganda in Africa. Genus is Orthohantavirus. It causes disease, hemorrhagic fever with renal syndrome. Name of disease is hemorrhagic fever with renal syndrome. Name of this virus, of this genus, was given due to the place of its discovery. It was discovered near Hantan River in South Korea. And another genus is Nairovirus that causes Crimean Congo hemorrhagic fever. That was named for Nairobi sheep disease virus. Nairobi sheep disease virus resembled this disease, but then this virus was isolated and studied, and that's why it was identified as another genus. But it resembled Nairobi sheep disease. That's why Nairo virus. What about Bunyavirida viruses? These are spherical viruses. Their genome is triple segmented. Look here, one segment, the second, the third segment. There is a small segment, medium segment, and large segment. A virion contains transcriptase, and there are four major polypeptides. This virus is envelope. What are antigens? It's nucleoprotein and glycoprotein. glycoprotein. This one. Cultivation is available in cell lines like HeLa and HEP2. Epidemiology and pathogenesis diseases caused by Bunya viruses. I have said that the first one is Orthohantavirus causes hemorrhagic fever with renal syndrome. The source of hemorrhagic fever with renal syndrome are urban rats, only urban rats. Transmission are elementary with water and food containing rodent excreta. Airborne transmission through inhaling aerosols of rodent excreta, urine, feces, or saliva, and sometimes, that is rare, but it also may be, but through biting of rodents. Hemorrhagic fever with renal syndrome is an acute viral infection that causes interstitial nephritis that can lead to acute renal insufficiency and renal failure in severe forms of the disease. Generalized hemorrhage and shock may occur. It is called hemorrhagic fever because it's characterized by multiple hemorrhages. In a mild clinical form, called nephropathia epidemica, the nephritis generally resolves without hemorrhagic complications and fatalities are rare, less than 1% cases. Immunity is lifelong after disease. So it is characterized by multiplication of virus in kidneys, so it causes nephritis, renal failure in severe form, and generalized means multiple bleeding in internal organs and in mucosal membranes. Another disease that is caused by Bunya viruses, that is caused by nairoviruses, Crimean Congo hemorrhagic fever. The source of disease are wild animals like hares or rats, domestic animals like sheep, goats, and cattle. Transmission through tick bites, biting of ticks from these animals. Incubation period from 5 to 6 days. This disease is characterized by flu-like symptoms like fever, headache, muscle ache, signs of bleeding, mental confusion, throat rash, vomiting, black stools, black due to the presence of blood clotted with multiple clotting because there is bleeding in intestine. The liver becomes swollen and painful, kidney failure, and shock. Microbiological diagnosis, treatment and prophylaxis of diseases caused by Bunya viruses. Specimen includes collection blood and urine. Methods for diagnosis are PCR, direct detection of virus, cultivation of virus in tissues, and serological tests, for example, immunofluorescence test. What about treatment and prophylaxis? Hemorrhagic fever with renal syndrome is not prevented by vaccines. There is no vaccine for this disease. Treatment involves supportive therapy including renal dialysis due to renal disorder. And there is antiviral drug that is ribavirin. In Crimean Congo hemorrhagic fever, we also may use ribavirin for treatment and immunoglobulin injection. Vaccine is also absent. And the last group that we talk about today, these are filoviruses that cause African hemorrhagic fevers. Family Filoviridae, and there are two genera here. These are Marburg virus and Ebola virus. These viruses cause African hemorrhagic fevers. African hemorrhagic fevers. Viral hemorrhagic fevers. This term, viral hemorrhagic fevers, refers to a clinical syndrome characterized by acute onset of fever accompanied by non-specific findings of malaise, prostration, diarrhea, and headache. Patients frequently show signs of increased vascular permeability, and many develop bleeding diathesis. The hemorrhagic fevers viruses represent potential agents for biological warfare, biological weapon, because of capability of aerosol transmission. Aerosol means through airborne. This one of the easiest transmission way. High morbidity and mortality associated with infection, and ability to replicate in cell culture in high concentration. Virus replicates with production of large amount of viruses. The important affecting agents are Ebola and Marburg viruses. Firstly, about their morphology. Their morphology is very interesting. They are long filaments. Look here, long filaments. So they are called filoviruses. Philo means filament, or filoviruses are long filament-shaped viruses. The genome is a non-segmented single-stranded DNA. Single-stranded RNA that is negative. They have seven polypeptides on their surface. These are enveloped viruses. Their antigens are glycoprotein surface, and proteins. This one, for example. Cultivation is available in cell lines, vero cells. Firstly, about history of Ebola, and then about Marburg. Why we should know about its history? Because the last epidemic of Ebola was severe epidemic was in 2014, about 8 years ago, and it is present now in African countries. About Ebola. Ebola virus was discovered in 1976, so we can say that it's a new virus, young virus, with occurrence of two severe epidemics of hemorrhagic fever in Sudan and Zaire. Now it is called Democratic Republic of the Congo. The outbreaks involved more than 500 cases, and at least 400 deaths caused by clinical hemorrhagic fever. 500 cases and 400 of them died. It's almost 90% of died patients. In each outbreak, hospital staff became infected through close and prolonged contact with patients, their blood, or their excreta. Firstly, it was not known about transmission ways of Ebola. It was very difficult to understand. That's why almost all medical staff were infected too. The largest known Ebola outbreak is occurring in Western Africa in 2014, with over 10,000 deaths to date in Guinea, Liberia, and Sierra Leone in Central Africa. Despite international emergency response and quarantine measures, the outbreak has not been controlled, and there remains a risk of spread to other areas. After that, they thought that this this outbreak was stopped, but no, it was reoccurred in August in 2018, and is present up to this moment in Democratic Republic of the Congo and Uganda. patients, people in this country and other nearest countries are still infected every day. What about Marburg virus? Marburg virus were discovered earlier in 1967, first identified in lab workers exposed to tissues of African green monkeys imported to Germany and Yugoslavia. Actually, African green monkeys were important, were transferred to Germany in scientific institute for experiments with other viruses, but these monkeys were infected with a new unknown virus, and some lab workers were infected and died from new disease. After that, scientists started to study it. Transmission from patients to medical person occurred with high mortality. Now about epidemiology and pathogenesis of African hemorrhagic fevers. Ebola virus. Ebola is a natural reservoir in nature are fruit bats. Source may be sick men and animals. Fruit bats leave their excretions, especially saliva or feces, in caves, for example, and especially people, tourists who like to visit unusual places like wild caves and other places where fruit bats may live. They are infected in most cases. For Marburg, natural reservoir are African green monkeys. These are very, very beautiful monkeys, so beautiful. And transmission through direct contact with blood, secretions or tissues from affected patients or animals. Direct contact and airborne. It's considered that airborne is rarer than direct contact. So in nature it's African green monkeys, and it also may be transmitted from infected human through contact with human body fluids that contain this virus, blood, secretions or tissues. Airborne is also present, but it's rarer than direct contact. Incubation period is from 3 up to 10 days, 9 days for Marburg disease, and from 2 up to 21 days for Ebola. Virus enter from small lesions, small cuttings in, for example, skin, in mucosal membrane, spread to lymph nodes, liver, and adrenal glands. Then viruses replicates in cells, in lymph nodes, in liver, in internal organs. And there are some mechanisms of action of this virus on human organism. The first one, they activate neutrophils. Neutrophils are activated because there is inflammation process. Massive activation of neutrophils trigger systemic inflammatory response. Neutrophils release a lot of pro-inflammatory cytokines. Pro-inflammatory cause severe inflammation. It is manifested in fever, malaise, gastrointestinal symptoms, and possible sepsis. Another one, when macrophages and monocytes are activated by the presence of this virus, they produce such cytokines as tumor necrosis factor, interleukin-6, and tissue factors that cause endothelial leakage. Viruses multiply in liver cells, and it causes hemorrhages. Hemorrhages in internal organs due to destruction of endothelium. Dendritic cells. Dendritic cells, they present, as the macrophages too, they present their main function, I hope you remember, presentation of antigen to T cells and activation of T cells. But this virus affects dendritic cells. It causes premature contraction by apoptosis, and it leads to impaired immune response. Immune response is not possible. T cells are not activated. All these mechanisms cause to multi-organ failure and shock. Multi-organ failure and shock. In this photo you can see main clinical manifestations of African hemorrhagic fever. It's multiple bleeding in eyes, in skin, in all internal organs, and blood is released from ears, from mouth, from genital organs, from all organs, and it is characterized by multiple internal bleedings. That is very dangerous. It calls this finally. Microbiological diagnosis, treatment, and prophylaxis of African hemorrhagic fevers. Specimen includes collection of blood, human body fluids, infected fruit bats and primates, monkeys, objects that have been contaminated with the blood. We can use PCR for detection of virus due to cultivation, and serological tests, for example, ELISA. Treatment and prophylaxis. There are no specific antiviral drugs for treatment and prophylaxis of Ebola and Marburg viruses. Treatment is directed at maintaining renal function and electrolyte balance and combating hemorrhage and shock, if it is possible. If it is possible, if it is early phase of the disease. For prophylaxis of Ebola, there is Ebola recombinant vaccine, RVSV, the above. Its brand name is Ervebo. In lecture on immunology, I said about it. This vaccine is under experimental development, but it was already used in last outbreak in the Congo in 2018-2019. And there is a viral vector vaccine, Zabdenovan M vaccine for individuals who are 1 year, children and older. It was made less than 2 years ago. In Marburg, there is no vaccine, but candidate vaccines are under development. The best way to avoid catching the disease is by not traveling to areas where the virus is found. Healthcare workers can prevent infection by wearing masks, gloves, and goggles whenever they come into contact with people who may have Ebola and Marburg. So that's all about these viruses. That's all about our lecture. Thanks for your attention. Prepare this lecture for next class. Goodbye.